Protein. Proteins. Proteins consist of one or more polypeptide chains, and each of these polypeptide chains are made up of monomers, which are amino acids. There are about 20 different types of amino acids. The basic structure of an amino acid, it consists of an alpha carbon here. The alpha carbon is bonded to a carboxyl group. The alpha carbon is also bonded to an amino group, and the alpha carbon bonded to a hydrogen atom, and the alpha carbon is also bonded to an R group or a side chain. So we have 20 different types of amino acid. So the amino acids are different from one another based on these side chains. Okay, each amino acid has different side chains. So you should be able to know how to draw an amino acid. So let me just draw it for you. So you have the alpha carbon, and then the alpha carbon is connected to an R group. The R group can be hydroxyl, can be amino group, can be hydrocarbons, can have carboxyl group or amino group. It depends on the type of amino acids. The alpha carbon is also bonded to an amino group. So amino, the nitrogen is bonded to two hydrogens. The alpha carbon is also bonded to another hydrogen. The alpha carbon is bonded to a carboxyl group. Carboxyl group is C double bond O OH. Okay, so you should be able to know how to draw an amino acid. So it has an amino group, a carboxyl group. Okay, so the amino acid also have different types of side chains. The R groups of amino acids can vary in structure, charge, so the R group can have positive charge, can also have negative charge, or does not have any charge or neutral. For example, if the R group have an amino group, so the amino group can accept another hydrogen ion to become positively charged. Or if the side chain has a carboxyl group, COOH, so when the amino acid is in an aqueous solution, so this carboxyl group will be ionized to become COO negative. So amino acid can have negative charge, can have positive charge, or if the side chain has a hydrocarbon such as methyl CH3, so it will be neutral. So the R group also allows the amino acid to be attracted to water or not. Okay, so the R group will allow the amino acid to vary in the affinity with water. Okay, and the reactivity with other molecules. So for example, if the R group contains hydroxyl group, so this makes the amino acid attracted to water molecule and causes the amino acid to be able to dissolve in water. Okay, so there are 20 different types of amino acid which commonly found in proteins. Each is identified by the different R groups. Okay, so each of these 20 different amino acid will have 20 different R groups. Okay, so amino acids are grouped by the properties of the side chain. So our groups can be polar, can be non-polar, can be acidic or basic. If the R group has hydroxyl group, for example, or sulfhydric group, or this basically hydroxyl, it causes the amino acid to be polar. So a polar amino acid allows the amino acid to be able to dissolve in water. Non-polar amino acid, if the side chain contains, for example, hydrocarbons such as methyl group here. So amino acid can be acidic if the side chain contains carboxyl group. Okay, so when the amino acid ionizes, the carboxyl group releases hydrogen ions, become COO negative. So if the amino acid is basic, it means that the R group contains amino group. Okay, amino group originally it is NH2. When it is ionized, it becomes NH3 plus. Okay, so these are the 4 different types of amino acids based on the side chain. Either it is polar, non-polar, acidic, or basic. Okay, so let's look at the 4 different types, the 4 different groups of amino acids. For the first group of amino acid, which is non-polar amino acid, which is the side chain is non-polar. Okay, so non-polar amino acids, the property of the amino acid will be hydrophobic. Okay, because the side chain contains hydrocarbons. For example, here we have glycine. Glycine is the easiest one, where the R group is hydrogen atoms. Okay, hydrogen atom bonded to the alpha carbon. Okay, so we have glycine, short form as GLY or G, and then we have alanine with the methyl group here, and then we have valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and also proline. As you can see, all these non-polar amino acids, they are hydrophobic because they have hydrocarbons as the side chain. Okay, so for your level, you are not required to remember this structure. Okay, just for you to know how to identify the amino acid by looking at the side chains. Okay. So the second group of amino acids are amino acids with polar side chain. Okay, polar amino acids, because the side chains are polar, so this allows the amino acid to be hydrophilic or attracted to water. Okay, so this increases the solubility of the amino acid in water. Okay, because this amino acid is attracted to water molecule. So for examples of amino acid with polar side chains are serine, threonine, cysteine, tyrosine, asparagine, and glutamine. So as you can see, for this polar amino acid, they have hydroxyl. We know that hydroxyl is polar. It can form hydrogen bond with water molecule. So same to threonine, it has hydroxyl, sulfhydryl, and then tyrosine also have hydroxyl, and then asparagine, it has a carbonyl group, and then glutamine also have a carbonyl group. So by having these functional groups, so it allows the amino acid to be polar and hydrophilic. The next group of amino acid is acidic amino acid and basic amino acid. Okay, so it is the amino acid have electrically charged side chain. So because the side chain have charges, either positive or negative charge, so this allows the amino acid to be hydrophilic. Again, it can attract water molecule. Okay, so examples of acidic amino acid with negatively charged side chain are aspartic acid and glutamic acid. Okay, so as you observe, the side chain for acidic amino acid, it has carboxyl group. Okay, glutamic acid also have carboxyl group. So when ionized in water, originally the side chain have COOH, so when ionized it becomes COO negative, releasing hydrogen ion into the surrounding solution, causing the surrounding solution pH to drop, which is acidic. Okay, and then for the basic amino acid, this amino acid have positive charge, okay, due to the presence of amino group, okay, at the side chain. So we have amino groups on each of these basic amino acid. We have lysine, arginine, and also cysteine. So originally the amino group is NH2. So when ionized in water, it becomes NH3 plus. Okay, because this amino acid will take up hydrogen ion from the surrounding solution, causing the pH of the solution to increase. Okay, so because the excess hydrogen ion in the solution will form bonds with the amino acid to become NH3 plus. Okay, so the amino acid will have positive charge. Alright, so amino acids are amphoteric molecules. Amphoteric molecules are molecules that have both basic and acidic groups. As in the case of amino acid, it has both amino and carboxyl group. Okay, so when amino acid is ionized, the amino group, which is NH2, will become NH3 plus. The carboxyl group COOH will become COO negative. Okay, so amino group is an amphoteric molecule, can act as both acid and base. So in water, its amino group, which is base, is ionized to NH3 plus. So for example, here we have an amino acid. Okay, C double bond O, OH, and then hydrogen, and then a side chain, and then amino with 2 hydrogen bonded to it. Okay, so the hydrogen at the carboxyl group, the carboxyl group will be ionized, releasing the hydrogen atom at the carboxyl group, and this hydrogen atom will become hydrogen ion in the solution. Okay, in the solution, so it releases hydrogen ion into the solution. So this hydrogen ion will be accepted by the amino group. Okay, so this amino group will add the hydrogen ion from the solution. It becomes positively charged at the amino group region. Okay, so amino group is ionized to become NH3 plus. The carboxyl group is ionized to become COO negative. So once hydrogen atom is released from the carboxyl group becomes negatively charged. Okay, so molecules with amphoteric properties can function as pH buffer. Okay, pH buffer. So pH buffer allows it resists any changes. The molecule will resist any changes in pH and try to maintain the pH of the solution. Okay. Okay. So when amino acid is dissolved in water, the amino acid dissociates into to form a zwitter ion, okay, which is bipolar. So we know the amino group is ionized to become NH3 plus. The carboxyl group is ionized to become COO negative. Okay, so this is how you should draw the zwitter ion. So the carbon is bonded to a NR group, a hydrogen atom, and then the carboxyl group is ionized to become COO negative. The amino group is ionized to become NH3 plus. Okay, the plus sign should be at the nitrogen actually. Okay, at the nitrogen atom of the amino of the amino group. Okay, so the basic amino group will function as proton acceptor. Okay, proton acceptor. So the amino group will accept the hydrogen ion from the surrounding solution to become NH3 plus. Okay, the carboxyl group, the acidic carboxyl group, becomes a proton donor by releasing the hydrogen ion into the surrounding solution. Okay, proton donor is for the carboxyl group. It releases a hydrogen ion or a proton into the surrounding solution. So carboxyl group donates a proton to the amino group. Amino group will become a proton acceptor, okay, forming a zwitter ion in an internal acid-base reaction. So the carboxyl group releases hydrogen ion and it is donated to the amino group. Amino group is the proton acceptor. So zwitter ion will have both negative charge and also positive charge. Okay, so amino acids polymer. Amino acids are linked by peptide bonds in a polypeptide chain. So a polypeptide is a polymer of amino acids. Okay, so as we can see in this diagram here, it shows you the formation of a polypeptide. So we have here a dipeptide. So two amino acid, one, okay, one amino acid, and the other one, another amino acid, are linked together through the formation of a covalent bond. We call it as a peptide bond. So a peptide bond will link between the two amino acids. So another amino acid will be is going to be added to the dipeptide to form a polypeptide, a longer chain. Okay, so to add a new amino acid to the dipeptide to form a polymer is through the process of dehydration reaction, okay, or condensation reaction. So the reaction involves the removal of hydrogen atom from one amino acid, okay, the amino acid to be added, and the removal of OH from the carboxyl group of the initial amino acids, okay, amino acid in the chain. So the hydrogen atom and the hydroxyl group that is removed will combine together to produce a water molecule in the dehydration reaction. So one water molecule is discarded from the reaction to connect two amino acids together. Okay, so these will form a peptide bond between the two amino acids. Okay, a peptide bond is a type of covalent bond between the amino acid in a polypeptide chain. So the polypeptide chain will have a backbone consisting of a repetition of nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon. So this is the backbone of a polypeptide. So the protruding from the backbone, you have the side chains of the different amino acid that is attached together. Okay, so the polypeptide chain has two ends. The at this end it has the amino end or the N terminus. On the end of the polypeptide chain, on the other end, it has a carboxyl end or the C terminus. Okay, let's look at this example. Okay, so an easier way to understand how the reaction occurs to join one amino acid to another amino acid, you have to draw it yourself. Okay, so let's draw one amino acid so you know the basic structure. So you have the alpha carbon bonded to a side chain, bonded to a hydrogen atom, and then it is bonded to a carboxyl group, and then sorry, not the carboxyl group. On this side we have to have the N terminus. Okay, we have to have the N terminus, okay, which is on this side it has the amino group. Okay, so on this side we have the carboxyl group. Okay, so this is one amino acid 1. Okay, so now we have the second amino acid. The second amino acid is also the same. You draw the alpha carbon, the side chain, the hydrogen atom, the carboxyl group, okay, and then the amino group. Okay, so in order for you to join this, so this is the second amino acid 2. Okay, so the reaction involved here is dehydration reaction. Okay, dehydration reaction. Okay, so in this reaction, the amino group of the second amino acid, which is the amino acid to be added to the chain, the hydrogen atom will be removed, and then on the amino acid 1, the OH will be removed. These two atoms will combine together to form a water molecule. Okay, a water molecule. So once you have removed the hydrogen and the OH from these two amino acid, you have to connect the two amino acid to form the product, which is in this case the dipeptide. Okay, the dipeptide. So you join the two amino acids together. You just draw back the initial structure, which is you have the nitrogen with the two hydrogen, the carbon, the side chain, the hydrogen atoms, the O. Okay, so you have removed the OH from the carboxy group, so now you have to connect the first amino acid to the second amino acid through the formation of a peptide bond. So carbon will form bond with the nitrogen atom. Okay, so nitrogen, you have removed one hydrogen atom, so you just draw one hydrogen that is joined to the nitrogen. Okay, tak ada dua dah, satu je dekat nitrogen untuk yang ada hydrogen tu. So the carbon, the side chain, the H, and then the carboxy group. Okay, so I don't have enough space there. Okay, so it is the same. Alright, so this is whenever you were to answer this type of question, you have to label where exactly the peptide bond. So the peptide bond will be in between the carbon and the nitrogen. So label it peptide bond. Okay, peptide bond. So you just look carefully at the structure of the amino acid and draw it carefully. So remember not to add the atoms that you have removed. So there is no OH bonded to this C, there is no hydrogen bonded to the nitrogen anymore. So if you draw that, that will be incorrect. Your molecule, your product will be wrong. Okay, so for this dipeptide, okay, we have the N terminus, okay, we have the C terminus. Right, so that is how you draw the reaction to form a dipeptide, and applies for the formation applies the same for the formation of a polypeptide. Okay, so next is the classification of proteins. So proteins can also be classified based on these four ways of classifying proteins. The first one is level of organization. So we have the primary structure, secondary structure, tertiary, and also quaternary. And then protein can be classified based on its structure, whether it is fibrous or globular protein. And then the composition of the protein, whether you have other elements added to the protein, such as metal ions, for example, to the protein. And then based on the function of the protein. So protein can have many different functions, such as protein can function as enzyme, as hormones, as structural proteins, as transport protein. It has many different functions. Okay, so the first way of classifying protein is based on the level of organization of the protein. So there are four levels, which is the first one is primary structure. Primary structure is linear sequence of amino acids. So this is the primary structure of polypeptide. So you have amino acids, different amino acids that are joined together through the formation of peptide bonds between the amino acids. So this is the primary structure, okay, linear sequence of amino acids. And then after the linear sequence, the primary structure, from the primary structure, it becomes folded in or coiled into secondary structure. Okay, so secondary structure results from hydrogen bonding involving the backbone of the polypeptide chain. So remember, the polypeptide chain, it has a backbone, nitrogen, carbon, carbon, nitrogen, carbon, carbon. That is the backbone of a polypeptide. So this secondary structure, there are two types. It can be coiled into a alpha helix structure, or folded into a beta pleated sheet. Okay, so alpha helix, it is coiled. Okay, coiled. This one linear chain of polypeptide, to become secondary, it can be coiled into a helical structure, or folded. Okay, folded into beta pleated sheets. Okay, so from the secondary structure, it can fold to become a tertiary structure. This tertiary structure is the result due to the interaction between side chains of amino acids along the polypeptide chain. Okay, so that one we are going to look at in the explanation later. And then after the tertiary structure is the quaternary structure, which is you have two or more polypeptide chain that interact to form a functional protein. So interaction among multiple, okay, two or more, okay, two or more polypeptide chains, okay, to form a functional protein such as enzyme or hormone, for example. Okay, for the primary structure is simple linear sequence of amino acids joined by peptide bonds. Genetic codes in DNA molecule determine the amino acid sequence of the polypeptide. Okay, so this is just a simple structure of a polypeptide chain for the primary structure. So you have different amino acids that are connected together through peptide bonds, and for example, you have one polypeptide chain. So one polypeptide chain A, for example, and we have polypeptide chain B. Okay, so this polypeptide chain later it becomes a functional protein, such as, for example, polypeptide chain A will become enzyme A, polypeptide chain B becomes enzyme B. So each of these enzyme will have different function. Okay, different enzyme catalyzes different reaction, for example. Okay, so each of these enzyme is composed of a polypeptide chain of different sequence of amino acids. Okay, different sequence of amino acids. Enzyme A will have different sequence of amino acid compared to the enzyme B that have different sequence of amino acid in the polypeptide chain. So the sequence of this amino acid that later will become each of these enzyme is determined by the genetic code in your DNA. So your DNA is very large molecule. It is to store your genetic information. So this genetic information is stored in the form of genes. Okay, gene. So each gene will code for the synthesis of different proteins. Okay, we have gene 1, gene 2, gene 3, for example. Each of these gene will synthesize polypeptide chain with different amino acid sequence. Okay, so the second structure, the second level is the secondary structure. So for the secondary structure, segments of polypeptide chains are coiled or folded, which lead to the protein's overall shape. Okay, it coils and folds which is the results of hydrogen bonds that are formed between carboxyl group and also amino group of the polypeptide chain. Okay, so there are two types of secondary structure. We have the alpha helix, which is in the coiled form, and the beta pleated sheet, which is in the folded form. Okay, so let's look at the alpha helix structure. So the polypeptide chain will be coiled into a helical structure. This helical structure are held together by hydrogen bonds that are formed between the carboxyl group and amino group along the backbone of the polypeptide chain. So hydrogen bonds are formed between the carboxyl and also the amino group along this polypeptide chain. So alpha helix structure are held together by hydrogen bonds. Same goes to the beta pleated sheets, okay, where you have a strand of polypeptide chain, okay, a strand of polypeptide chain. The region that are parallel to one another will be connected through the formation of hydrogen bonds also. So this strand of polypeptide will be folded, and the folded region that are parallel to one another will be held together by hydrogen bonds between the carboxyl and the amino group along the polypeptide chain's backbone. Okay, so the helical structure, the alpha helix structure, the polypeptide chain is coiled to form a helical structure. So the helical shape is maintained by hydrogen bonds between an oxygen of a carboxyl group, okay, and also an oxygen of carboxyl group of one amino acid, okay, of one amino acid, such as for example this one here. This is one amino acid, okay, and the hydrogen of amino group of the fourth amino acid along the chain, okay. So this is one amino acid, this is another amino acid, so it is every 4 amino acid down the chain, okay. 1 2 3 4. So every 4 amino acid there will be formation of hydrogen bonds between the carboxyl and the amino group of every 4 amino acids along the polypeptide chain, okay. So the interaction is through the formation of hydrogen bonds. Okay, so the hydrogen bonds will hold the helical structure of alpha helix. Okay, so the second type of secondary structure is beta pleated sheets. Okay, so just look at this keyword. It says here 2 or more segments of a single polypeptide chain are arranged parallel to each other and are held by hydrogen bonds. Okay, just look at the diagram carefully. For secondary for both secondary structure, be it alpha or beta, okay, alpha or beta, it only involves one polypeptide chain, okay, one polypeptide chain. So same goes to the beta pleated sheets. One polypeptide chain that are folded together, like this one here, okay, that are folded, okay, from one polypeptide. They are from one polypeptide chain, it is folded, and the segment that are parallel to one another will be folded, okay, will be folded, and the folded region here will be held together by hydrogen bonds, okay, by hydrogen bonds also, okay. The segments that are parallel to each other are held by hydrogen bonds. Hydrogen bonds hold neighboring strand in different regions of a polypeptide chain. Okay, it only involves one polypeptide chain that are folded, and the segments that are parallel will be folded and hold together by hydrogen bonds. So the polypeptide chains become folded, and this type of structure, which is the beta pleated sheets, has high resistance to stretching, and it is the beta pleated sheets is strong and flexible. Tertiary structure. Tertiary structure is the overall 3D shape of a polypeptide chain that results from interactions between the R group of amino acids along the polypeptide chain. So the interactions between the side chain is the result from hydrogen bonding. It results in hydrogen bonding, ionic bonds, or hydrophobic interactions, or van der Waals interactions. Okay, so strong covalent bonds called disulfide bridge may reinforce the protein structure. So basically, the tertiary structure is results from one polypeptide chain that is folded, okay, it is folded into a specific shape due to the interaction between the side chain of amino acids along the polypeptide chain. Okay, so it is due to the interaction between the R groups that forms hydrogen bonds, ionic hydrophobic interaction, and van der Waals interaction between the side chains. Okay, so example given here is to show you how the side chains along the polypeptide chains are formed. Okay, so we have um, just to draw to you, okay, the amino acids along the polypeptide chain. So the purple strands here is it indicates actually the backbone of the polypeptide chain, consisting of nitrogen, carbon, carbon. So you have the amino and the N terminus and also the C terminus. Okay, so the amino acids are grouped based on the side chains. Okay, so for example here, these side chains, this amino acid have side chain that is polar. It has hydroxyl group. Okay, polar because it has hydroxyl group. Along the polypeptide chain, it meets with another amino acids, okay, that also have polar side chain. It has a carbonyl group. Okay, a carbonyl group has C double bond O. It has oxygen. So the oxygen and the hydrogen of the hydroxyl group of the amino acid, it will form hydrogen bonds. Okay, hydrogen bond. So this is the interaction that is formed between the side chain of the amino acids along the side chain, along the polypeptide chain. Okay, so you have another amino acid. Okay, another amino acid here, where the amino acid have hydrocarbons. Okay, so this is the amino acid that have hydrocarbons will attract another amino acid that also have hydrocarbons as the side chain. So the side chain, the two amino acids side chains interact with each other to form hydrophobic interactions and also van der Waals interaction. So this is to keep the polypeptide chain folded into a specific shape. It is folded into a specific shape, which is the tertiary structure here. Okay, and then along the chain again, we can see there's another amino acid that has a sulfur. Okay, it has a sulfhydryl group, SH. Okay, SH, a sulfhydryl group. Okay, a sulfhydryl group. Sulfhydryl, and then another amino acid also have a sulfhydryl group. Okay, for example, cysteine. So the sulfhydryl group will catalyze, the amino group will involve in a chemical reaction, so that a disulfide bridge, a covalent bond, is formed between the sulfur and the hydrogen atom in the sulfhydryl group will be removed to allow the sulfur to form a covalent bond with one another to form a disulfide bridge. So disulfide bridge is a covalent bond. It is quite a strong bond. It is a strong bond. So this keep the polypeptide chain to be folded in this specific shape. Okay, and then along the chain again, you see another amino acids. Okay, amino acids with amino group that has positive charge, and then also another amino acid that have a carboxyl group that have negative charge. So oppositely charged side chains of these two amino acids will be attracted to one another through the formation of ionic bonds. Okay, so for the tertiary structure, it is basically from one polypeptide chain that is folded into a specific shape, and then due to the interaction between the side chain of amino acids along the polypeptide chain, due to hydrogen bonding, disulfide bridge, hydrophobic interaction, van der Waals interaction, and also ionic bond. The fourth level is quaternary structure. Quaternary structure results when two or more polypeptide chains combine together to form a protein molecule with specific shape and function. Okay, so example of protein that has quaternary structure are collagen. Okay, so collagen is a fibrous protein consisting of 3 polypeptide chains that are coiled like a rope. And then the second example is hemoglobin. Okay, hemoglobin is a type of globular protein. It folds into a specific shape. Okay, and the hemoglobin consisting of 4 polypeptide chains. Okay, 2 polypeptide of type alpha, and the other two is the other two polypeptide is of beta type. Okay, 2 beta chain. Okay, so as you can see, this is the structure of a collagen. So it consists of 3 polypeptide chains that are coiled around together. Okay, that are coiled around together. So collagen is a type of fibrous protein. It will not dissolve in water. Okay. The second type here, the next one is hemoglobin. Okay, hemoglobin is a type of globular protein. Okay, globular protein. So this globular protein, the hemoglobin, is consists of 4 polypeptide chain. Okay, so quaternary structure of protein, it consists of 2 or more polypeptide chains that are combined together to form a functional protein. Okay, so hemoglobin here, it has 2 alpha chain and 2 beta. Okay, 2 beta chain. Okay, 2 beta subunit and 2 alpha subunit to form a functional hemoglobin. Hemoglobin is the pigment in red blood cells that functions to carry oxygen. So each of these subunit, okay, consists of a polypeptide chain with a hem group. Okay, a hem group, where the hem group it has an ion. Okay, a ferrum. Okay, a ferrum. So the function of the ion or the ferrum in the hem group is to bind to oxygen molecule. So one hemoglobin can bind with 4 oxygen molecule. Okay, so you can also classify protein based on the structure of the protein. So protein can be classified in 2 forms, okay, which is as either fibrous protein or globular protein. Okay, so for fibrous protein, it is consists of long parallel polypeptide chains. Okay, long parallel polypeptide chain. As for the globular protein, it is coiled and folded into a globular shape. Okay, like in the tertiary structure that we have looked before. Okay, so you have one polypeptide chain that folds into a specific shape. Okay, so the one that we have an example just now is the hemoglobin. Okay, the polypeptide chain are coiled and folded into a specific shape. Okay, so for the that is the first difference. The second difference, for the fibrous protein, polypeptide chain form helical structure or pleated sheets that are held by hydrogen bonds, like the one that we have just looked at for the collagen. So the collagen consists of 3 polypeptide chain, and each of the polypeptide chain is in the form of alpha helix, and these alpha helix are held together by hydrogen bonds. Okay, the polypeptide chains form helical structure or pleated sheets held by hydrogen bond. As for the globular protein, the globular structure of the protein are held together by the interactions between the side chain, okay, between the side chain through the formation of hydrogen bonds, ionic bonds, or hydrophobic interaction or disulfide bridge, okay, which causes the polypeptide chain to attain a certain functional shape. Okay, and then the third one The third difference between fibrous and globular is that for fibrous, it is composed of secondary structure, okay, secondary structure as either helical or pleated, okay, pleated along a polypeptide chain. So it is important in carrying its function as structural and supporting protein. So for example, collagen or keratin that are found in skin or hair, it is made up of protein that is fibrous, and it cannot dissolve in water. Okay, it cannot dissolve in water. It is insoluble in water. And as for globular protein, it is composed of protein is in the tertiary or quaternary structure, which is important in determining its metabolic function. So globular protein, they are usually soluble in water and can form colloidal suspension. Example of globular protein, we have hemoglobin, we have enzyme, we have hormones. All these molecules are carried in your bloodstream. Major component of your blood is water. Okay, so all these molecules, enzyme, hormones, enzyme, hormone, hemoglobin, they are soluble in water. Okay, so this is to allow these molecules to be carried in your bloodstream. And then the next comparison between fibrous and globular protein. For fibrous protein, the amino acid sequence along the polypeptide chain is regular, repetitive and regular, repetitive, regular amino acid sequence. Okay. And then for the globular protein, the arrangement of the amino acid is irregular. So we have uh, so many different types of enzyme, so many different types of hormones. So all these hormones and enzyme have different functions. So therefore, the sequence of the amino acid will be different, irregular, from one protein molecule to another protein molecule. Okay. And then example of fibrous protein is we have collagen. Collagen is the protein that are found in your skin, in your nails, in your hair. And then collagen, and then you have elastin, fibrin in silk protein. Example of globular protein: hemoglobin, myoglobin, antibody, and enzymes. Okay, so the third method of classifying proteins is through the composition of the protein. So, if the protein are made up of only amino acids, it is called as simple protein. It consists of only amino acids, do not contain any other substance such as metal ions or any other substance. It is mainly made up of amino acids. Examples are albumins, okay, which is egg albumin, serum albumin. And then we have globulins such as antibodies and fibrinogen. And then we have histone, which is protein associated with DNA. So your DNA is a very long molecule. In order to fit into a small nucleus, so this DNA molecule will be coiled around a protein. We call it as protein histones. Okay, so the protein will be coiled around uh, a histone protein, okay, to make the DNA fit into the nucleus in the form of chromosome. And then we have scleroprotein, which is keratin, collagen, and collagen in connective tissues. The second type of protein based on its composition is conjugated proteins. Conjugated proteins are proteins with other non-protein component. Okay, it has amino acid obviously, and also non-protein component or materials known as prosthetic group. Okay, this prosthetic group, the presence of the prosthetic group in the protein is crucial for the function of the protein. Okay, so example of conjugated protein are lipoprotein, glycoprotein, myoglobin, hemoglobin, and nucleoprotein. For lipoprotein, it is the prosthetic group is lipid. Okay, so it is the component of cell membrane. Okay, lipoprotein is one of the component of cell membrane. And then we have glycoprotein, okay, which is the combination between protein and polysaccharides, okay, which is also found in the membrane, embedded in the plasma membrane and blood serum. And then myoglobin, the prosthetic group is Hem group, which is found in muscle cell. And then hemoglobin, the prosthetic group is Hem, which contains iron. So both hemoglobin and myoglobin contains Hem, that contains iron. Okay, so hemoglobin is the pigment that is found in red blood cells or erythrocyte, which is the iron in the hemoglobin is used to bind with oxygen. So hemoglobin without a Hem group or iron, so it is not functional as it cannot bind to oxygen. So hemoglobin has to have Hem group that has iron to bind to oxygen, to carry oxygen molecule. And then the last one is nucleoprotein, found in nucleic acid. It is the combination between protein and nucleic acids, okay, the histone just now, to form a chromosome, and also ribosome, the component within ribosome. Okay, and the last way of classifying the protein is by looking at the function of the protein. So there are so many different proteins with different functions. Okay, so there are proteins that have structural functions, such as, for example, keratins, which is the protein that are found in your hair, in your nail, in scales, in feathers and horns, and also silk. Forms web and also cocoon. So structural proteins. So these protein that form structures, they are insoluble in water. And then there are protein that involves in movement, such as, for example, actin and myosin in muscle contraction. Okay, in muscle contraction allows the muscle to contract. And then defense, okay, such as antibodies, okay, which is found in your blood, which is to fight against pathogen, okay, pathogen. So the antibody will bind to foreign substance or virus or pathogen that enters into your body, and it prevents you from getting sick from these pathogens. Okay, and then we have protein that have storage as it function. Okay, so such as albumin, okay, albumin in egg white, which provide nutrition for embryo in egg. And then there are protein that function in signaling, such as, for example, insulin. So insulin is hormone, so there are hormones that are made up of proteins function in signaling, which coordinates your body functions. Okay, insulins, it controls your blood sugar levels. Okay, and then the last one, the function of protein. There are protein that function as enzyme to catalyze chemical reactions, such as amylase, which is the enzyme found in saliva and also in the small intestine to digest carbohydrates. Okay, so the last part of protein is denaturation of protein. Protein on denaturation. So protein, because it attains specific shape, okay, a specific shape, and this specific shape of protein allows the protein to carry out its function. Okay, so and this specific shape of protein, as we have looked before, some of it are held by hydrogen bonds. Okay, hydrogen bonds between the side chain. Okay, so as we know, hydrogen bonds, it is easily broken due to heat. So if you insert if you insert heat to a sample of protein, so this can cause the protein to lose its shape due to the hydrogen bonds that breaks between the side chain, and this causes the protein to denature. Okay, so protein on denaturation, loss of a protein's original structure or 3D shape due to the change in pH. These are the factors that cause protein to denature: pH, salt concentration, and also temperature. Okay, so if you increase temperature, you can cause the hydrogen bonds between the side chain to break. If you change the pH of a solution, for example, if you add acids into a solution containing protein. So you have excess of hydrogen ions. So this excess of hydrogen ion will disrupt the ionic bonds, okay, the ionic bonds between the side chains that interact with one another through ionic interaction, ionic bonds. Okay, so you changing the pH of the solution will mess with the ionic bonds between the side chain and causing the protein to lose its shape. Okay, which is the protein now will be denatured. Okay, so example here you have a normal protein that folds into a specific shape. By folding into a specific shape, this protein can function normally as enzyme or as hormone, for example. And then if you change the pH, you change the salt concentration, you change the temperature, the bonding between the side chain will be disrupted. Okay, the hydrogen bonds, the ionic bonds, the van der Waals interaction will be disrupted. Okay, so and causes the protein to denature. So a denatured protein sometimes can return to its functional shape when the denaturing agent is removed. Okay, um, uh, you uh, you change the pH back to the normal pH, for example. So and then the chemical and the physical aspect of its environment are restored to normal. Okay, so sometimes you can reverse the reaction. The denatured protein can be reversed back to its original shape. Okay, sometimes. Okay, so that's the end of proteins. So thank you.